Okay, so welcome to the Alpha Therapy session. I'm Kalevi Kairemo from Helsinki, Finland, and we have 5 speakers in this session. So Professor Hatazawa and Watabe from Osaka, Japan, will speak about astatine 211 in thyroid cancer, and then Professor Bruland and Asta Juzeniene from Oslo, Norway, will talk about dual targeting of prostate cancer in PSMA-positive prostate cancer cells. Next. Professor Hatazawa comes from Osaka, and he's there, the director of Department of Nuclear Medicine, University Hospital, and University Positron Emission Tomography Imaging Center, and he's also the president of Asia Oceania Federation of Nuclear Medicine and Biology from 2017 to 2020, and his research interests deal with diagnostic imaging and radionuclide therapy, and especially alpha therapy and boron neutron cation therapy. Next, please. Øyvind Bruland is a professor of clinical oncology and Faculty of Medicine in University of Oslo. He has published more than 150 papers in international journal and written several editorials and reviews and book chapter. And he is the founder of Algeta, who actually developed radium-223, which is currently marketed as Xofigo, and he's also co-founder of Nordic Nanovector, which develops alpha-labeled monoclonal antibodies. I will speak about current practice in alpha therapy. Next, please. So the combination of diagnosis and therapy is entitled also diagnostics. So it's meaning that we use diagnostic radionuclides and replace them with therapeutic radionuclides, and we can use to characterize the process in vivo in patients, and then by switching suitable radionuclide we can treat these patients, and radiopharmaceuticals are prime examples of theranostics. Next, please. I won't speak about PSMA targeted alpha radium therapy, but just two examples. So actinium-225 is currently used in clinical testing as shown here, and the responses are remarkable, as seen on those gallium PET images. Next slide, please. Next. And similarly, another alpha emitter, bismuth 213, can be used for treating prostate cancer, as shown here. So it's complete metabolic response. Next. So I will focus on targeted alpha therapies in the clinical development. This is a busy slide. It deals applications with actinium-225. So there are cell differentiation antigen CD33 related trials, which has been used for AML, acute myeloid leukemia, then multiple myeloma, myelodysplastic syndrome, and ablation before ablation of bone marrow transplantation. There are also other application for CD45 for acute myeloid leukemia, and then there are peptide receptor studies where both lead and actinium labeled alpha isotopes have been used. Next, please. In a more detailed manner, here the difference of alpha particles and beta particles are shown. So the linear energy transfer is high with alpha particle, low with beta particle, and typically alpha particle causes double strand DNA breaks and cluster DNA damage, whereas beta particles are causing single strand DNA breaks. So a likelihood for DNA repair is low, whereas it is high with beta particles, and the penetration range is micrometers with alpha particles, whereas it's millimeters with beta particles, and range in cell diameters is less than 10, and and in beta particles reach up to 1,000 cells, and also the number of hits for cell killing, what is required is one-digit number with alpha particles, and you need several thousands when you talk with beta particles. Range in the air is less than 5 centimeters with alpha particle and few meters with beta particles, and alpha particle can be stopped with paper, whereas aluminium is required for beta particles, and crossfire effect is typical for beta emitting radiopharmaceuticals, and short range alpha emitters may induce limited hematotoxicity if the targeting is excellent. And on the right you see an example of actinium labeled. So typically it's called lintuxumab, the antibody what is used in hematologic malignancy. These are currently in clinical trials. Next. Another example is the lead-212, what is in clinical trials and development. These are European companies, so Oranomed is French company, and Nordic Nanovector is a Norwegian company. So example of lead labeled, it's in somatostatin receptor peptide therapy, and then in hematologic malignancy anti-CD37, that's another cell differentiation antigen is developed for clinical trials. Next. So the only approved radiopharmaceutical used for alpha therapies is radium dichloride. Next. And in Europe, there were actually three radiopharmaceuticals used for bone pain palliation. There were strontium chloride, samarium lexitronate, and rhenium 186 etidronate, and then before radium 223 got marketing approval in 2013. Next. So the radionuclides, strontium, samarium, and rhenium for bone pain palliation. So the indication is that the patient has to have bone pain and at least one side with an osteoblastic response on bone scan or sodium fluoride PET. Next, please. And the results with these, typically the bone pain palliation duration is up to 24 weeks. Next. And the targeting mechanism is shown here in the upper image, so that the bone therapy, so that you are targeting, this is thyroid cancer example, so you are targeting normal bone, reactive bone surrounding the metastasis, and whereas the tumor therapy, so these are not targeting the tumor cells per se, but reactive bone cells, osteoblastic bone forming cells. Next, please. So the registration trial was published in New England Journal of Medicine 2013 by the Parker and Stenilson, and you can see the survival curves on the left, and also the disease free time. So it was up to 77 weeks. If you remember that with beta emitters it was up to 24 weeks. Next, please. And we also developed in European multi-center European-based study how clinical centers should be optimized for using radium. And one of the elements is to detect bone metastasis either with bone scan or sodium fluoride PET. And I'm gonna briefly describe this sodium fluoride PET and its use in the next slides. Next, please. So I worked with the single institution trial at MD Anderson, published this 161 patient and more than 770 cycles. So the median survival alone was more than a year, 12.4 months. Next. And these are people from MD Anderson. Next, please. And we also studied the possibility of interim PET. So the radium therapy consists of 6 cycles of radium administrations, and here we did the PET before, after 3 cycles and 6 cycles, and we didn't found any systemic phenomenon which should be applicable for clinical practice. Next, please. Similarly, I studied in Helsinki sodium fluoride PET after the first cycle, and then after the 6th cycle, and also the PSA response, and as you see from these 10 patients which are shown here, so 9 out of 10 responded to the therapy. Next, please. And we also look at the early response after the first cycle, and then after the 6 cycles, and we actually got the same finding that you cannot predict the response on the base of sodium fluoride scan, but you can measure the outcome of the sodium fluoride of the radium 223 therapy, and by using sodium fluoride PET. Next, please. And here are some practical examples. This is a patient who didn't respond, not on the left there's a scan before therapy, and then 1 month later, so the uptake increased, but you cannot see this with visually. Next, please. And then when you have a super scan, this is an example, actually a patient who responded to therapy, so you cannot predict or detect this visually, but with quantitative analysis, you see that actually this patient responded that from the biomarkers, alkaline phosphatase might be sometimes more precise of assessing bone as the PSA marker per se. Next, please. Another example with small response. These are after one cycle and then after 6 cycles. They are on different scale, but when you do this quantitatively, so you see this difference. Next, please. And this is a patient also who responded, but you cannot see those with the naked eye, so you have to do quantitative analysis. Next, please. And this is the patient where you first before and then after the treatment there was a clear response. This was also quantitatively a remarkable response, so almost 70%. Next, please. So from this we can say that you cannot after the first cycle you cannot predict what the outcome will be, but on the other hand you can see the final response by using sodium fluoride PET, and also on the other hand these were some of the first patients, so that this was actually excellent therapy. Next. We also because this is a radionuclide therapy, you should develop. It's actually European law and legislation. You should have a therapy and dosimetry. It's actually mandatory, and this is we did some attempts to calculate dose in these kind of therapies. Next. And as mentioned, so the gamma component is about 1% of the radiation, and we did imaging from 0 to 8 days, and you see that the quality of the images is not that poor, but good, but you can do quantitative analysis. And next, please. And also here shown the patient with response or other very busy slide with all other therapies, and also the markers PSA and alkaline phosphatase, how they behave, and also that the patient, both markers responded to the therapy. Next, please. And then another patient who didn't respond to alpha therapy, but we studied this thoroughly. Next. And this is the data from gamma imaging. So we had a relative institute half-life, and then we can get the relative absorbed dose, and we see that there were just a few metastases, so then there is already huge variation in the relative absorbed radiation dose. Next, please. And we have summarized here the lesion half-lives, and then the also the relative absorbed radiation dose. So there was actually 4-fold difference in just a few metastases. So this means that the different bone metastases get different dose, even though the treatment regimen and then the patient sizes are somewhat similar. Next, please. And our results suggested that dose planning and calculus should be included as part of this treatment modality, because already in these patients the behavior is individual. But gamma imaging response cannot be predicted, because they had very similar doses, and not all patients responded. Next. So this is the summary of what I presented, the final outcome in a large population, the half-lives, and then the dosimetric things, and you basically should use bone scan or sodium fluoride PET in the follow-up, but interim studies are not useful. Next, please. So then I was involved with the phase 1 dose escalation trial with radium 223 in high-risk osteosarcoma. So we published this paper in clinical cancer research last year, and actually the main findings were that the osteosarcoma should be administered at least double the amount what is used for metastatic prostate cancer, and radium chloride exhibits signals from activity from calcified bony lesions, and it's also like brain metastasis or lung metastasis, but like samarium EDTMP, which I mentioned before, so that's myeloablative, but radium 223 is not. Next, please. So because of that development, so I actually developed new criteria for sodium fluoride, how you could evaluate response based on sodium fluoride PET, because all the tumors and lesions are sodium fluoride positive in bone tumors, primary bone tumors, and so you can create similar NAFSIS criteria. Of course, the patient population is small, but they worked in our setting, and also I published a book about the sodium fluoride PET in clinicals used together with Homer Makapinla from MD Anderson Cancer Center. Next, please. And so there are RECIST criteria are for CT and MR. PERSIST are for FDG PET, and NAFSIST are for sodium fluoride PET, but it's limited to sodium fluoride avid disease. Next, please. So these are also called Kairemo criteria, and then this is from the actually the Norwegian speaker, Professor Bruland. So his doc work more than 15 years ago in that you really target for micro autoradiography, that you really target the osteoblastic zone in sarcomas. Next, please. And actually the first presentation was when I was in Uppsala. I worked there 5 years, but this is more than 15 years ago. So the first ever sodium radium presentation was given by the inventor Roy Larsen in my inauguration seminar. So we have been the Scandinavians have been very long active in this field, and actually the Norwegians and Swedes developed this tracer. Next, please. And we had Warmth Congress 2012. So we were ahead of time. So there were Sten Nilsson and Roy Larsen and Øyvind Bruland were already awarded there in the Lapland Congress. Next. And there were a lot of people in Lapland that time try to enjoy the midnight actually not me aurora, northern lights, and other interesting things, aurora borealis, and a lot of snow. Next, please. And then I hosted 2018 the another World Congress in Helsinki, and then there was also discussion about another theranostics was developed and discovered for thyroid, both benign thyroid disease and then for malignant bone disease. Next, please. So the final slide of my talk. So the alpha therapy in the clinic. So radium 223 dichloride, also known as Xofigo, is ideal for metastatic castration resistant prostate cancer with bone metastasis. It improves overall survival. Early interim imaging does not predict the outcome, and response evaluation can be done with sodium fluoride PET imaging, as I told here, and or bone scintigraphy, and or then the markers with alkaline phosphatase or PSA. Dosimetry is cumbersome. Breast cancer alpha therapy might work in selected indications. There is anecdotal evidence-based medicine. Osteosarcoma, as I showed here, there is no evidence-based medicine yet, maybe in selected indications. And then there are other compounds under development, which I showed in the early some busy slides, but especially in hematologic malignancies. So I thank you very much for your attention. Thank you.
Hello, my name is Tadashi Watabe. I'm Assistant Professor of Department of Nuclear Medicine and Tracer Kinetics, Osaka University. So today I'd like to talk about astatine 211 preclinical study for the approval of clinical use in patients with intractable thyroid cancers. So first of all, I'd like to explain about astatine. Astatine is an alpha-emitting radionuclide. Its half-life is 7.2 hours. And in the periodic table, it locates just under the iodine. And it shows very similar similar properties to iodine. And it can be manufactured with an accelerator. And in clinical practice, iodine-131 treatment is performed for all thyroid cancer patients. And here is a representative case of lung metastasis of papillary thyroid cancer. And this patient showed a multiple lung metastasis. You can see very high uptake in this iodine image. And patient was treated with iodine-131 twice. And patient showed response to this therapy. But in another patient, this patient also shows multiple lung metastasis, and there is still some neck remnant lesion. But this patient did not respond to iodine-131 therapy. It showed worsening of lung metastasis, and the lymph node metastasis was also appeared. So we need more effective and strong treatment for this kind of refractory patient. So now we are focusing on astatine. And in this table, we compared between iodine and astatine. And of course, iodine emits beta ray, and astatine emits alpha ray. And astatine has more biological effect and also a more therapeutic effect can be expected. And another advantage of astatine is it shows very limited gamma ray. Actually, it is X-ray. And injected dose is much smaller than iodine. So usually in Japan, we need hospitalization in a dedicated room for iodine 131 high dose therapy. But for astatine, we don't need such hospitalization. So outpatient treatment is possible in astatine. And also the astatine can be produced with the accelerator. So domestic supply is also possible. So we started from the to review the chemical oxidative status of astatine at first, and just after the separation from the bismuth target from the accelerator, the astatine crude solution shows multiple components in this TLC analysis. And it is considered to be the astatine is composed of many types of oxidative status, AT minus and ATOH minus, or blah blah blah. And when we added ascorbic acid, these components can be converted into one. So this is considered to be AT minus. From the iodine 123 TLC analysis. And ascorbic acid shows the best performance among other reducing agents. And then we first injected this sodium acetate with ascorbic acid treatment into normal rats. And with the addition of ascorbic acid, the accumulation in the thyroid gland was increased about 3 times. And this uptake is gastric uptake. It shows almost similar between ascorbic added and no added group. And later, we performed a cellular uptake analysis using the human papillary thyroid cancer cell line. And left figure shows the comparison of uptake between K1 and K1 NIS. And without NIS, sodium iodine syn-porter function, the astatine was not taken up into K1 cells. So astatine was transported via NIS with this figure. And right figure shows the effect of ascorbic acid in the thyroid cancer cells. And with ascorbic acid, it shows higher accumulation, like normal thyroid gland. And then we injected this astatine sodium acetate into mice, xenograft mice, and it shows very high uptake at 3 hours and even at 24 hours after injection. And for the treatment effect, the control mice showed continuous growth of tumor, but astatine injected group showed dose-dependent treatment effect. 0.1, 0.4, and 1 megabecquerel injection. So these groups showed dose-dependent tumor growth inhibition. And for the body weight change, 1 megabecquerel injection group showed transient decrease of body weight, but it was recovered, and this group showed the best body weight maintenance because the tumor growth was the slowest. And we performed immunohistochemical staining using NIS antibody. And K1 NIS xenograft, of course, shows high expression of NIS. And also the thyroid gland and stomach shows physiological expression of NIS. So the astatine uptake reflects this kind of NIS expression. And we also performed a comparison between iodine and astatine. And for iodine, it shows tumor growth inhibition compared to control. But when we compare to astatine, this square dot, the treatment effect is higher compared to astatine, as compared to iodine, sorry. And we also performed a double-strand break assay in K1 NIS cell line. This is a human papillary cell line, which we used in the preclinical study. And you can see that those dependent increase of double-strand DNA double-strand break. And of course, astatine shows higher double-strand break compared to iodine. And the very interesting finding is if you can look at this astatine image, the double-strand break signal is almost saturated in the cells. But in iodine, the dose is 10 times. But you can see many spot-like signal. So it shows that double-strand break formation is still mild to moderate compared to astatine. And it reflects tumor growth inhibition in the xenograft model. So we expect this kind of higher therapeutic effect in the refractory thyroid cancer patients. And we performed the evaluation examination about side effects. And first, we evaluated the blood test examination, and the white blood cell count and also platelet count showed transient decrease at 7 days after injection, but it was recovered at 2 weeks after injection. And there is no significant change in the renal function or liver function. But in the late phase, so it was later than 2 weeks after injection, total cholesterol, albumin, and total protein showed increase in the high dose group. And it was considered to be the effect of thyroid aberration because we use normal mice in this research. And for histological analysis, in the low dose 0.1 megabecquerel group showed no significant change in the major organs, but high dose group showed some change. For example, thyroid gland shows the decrease of follicles, and you can see some inflammatory reactions. This is really aberration effect. And for testis showed mild accumulation of astatine. This is physiological accumulation, and it shows the transient decrease of sperm formation at 15 days after injection. But this is transient effect, and it was recovered. And for other risk organs, salivary gland, stomach, spleen, and kidney, they are within normal findings. And now we are preparing to start clinical trial using sodium acetate for thyroid cancer patients. And we had a consultation to Japanese authority for drug approval, PMDA, and we agreed to just to perform toxicity test in mice to start clinical trial in humans. And we performed the toxicity test and the reliability standard in Osaka University. So I will show you the results. So we performed extended single intravenous dose toxicity study using sodium aside, and we injected 3 kinds of dose of sodium aside, 5, 20, and 50 megabecquerel per kilogram of astatine. And we evaluated at 5 days and 40 days after administration. And we checked the general condition, weight, organ weight, blood test, and histopathological examination. So I will show you the results. And in the blood test result, this is male result, we observed a decrease of white blood cell and platelet counts at 20 and 50 megabecquerel per kilogram injection. This is a kind of mere suppression, bone marrow suppression. But this decrease was improved, recovered at 14 days after injection. This is a very compatible result as our preclinical study. And in females, it shows similar white blood cell counts and platelet counts reduction. And it also showed recovery at 14 days after injection. And for total protein, albumin, and total cholesterol, it shows increase in the high dose group at 14 days after injection. It is also compatible with our preclinical study, and female also shows the same trend. So we don't observe any additional abnormal findings when compared to the previous our preclinical study. And for organ weight, the spleen showed a weight decrease in the high dose group, and in high dose group also shows the salivary gland and kidney, testis weight decrease at 14 days after injection. And in female, it shows similar salivary gland, spleen showed decrease at day 5. And in high dose group, it also shows salivary gland weight decrease. So these are risk organs with mild to moderate accumulation of astatine, like iodine 131. And ovaries also showed the decrease of weight at the 40 days after injection. So we observed that kind of weight decrease in the high dose group, but most important examination is a histopathological examination. So I will show you these results. And in the thyroid gland, of course, thyroid is a target organ, and it shows colloidal decrease, and also infiltration of inflammatory cells were observed. So this is also compatible with our preclinical research study. And in testis, it showed multinucleated giant cells at 14 days after injection. So we observed this finding in all groups in high dose. So this is considered to be the effect of astatine. But for other organs, including risk organs such as salivary glands, stomach, spleen, and kidney, ovaries, it did not show any significant abnormal change in this toxicity study. So we conclude that the significant change was observed in the thyroid gland and testis only. So here is the summary. In the extended single dose intravenous toxicity study conducted under reliability standard, no abnormal finding exceeding the expectations of previous studies was observed. So the ablation effect of sodium acetate on the thyroid gland is already known, and it is not a problem because refractory patients have already undergone total thyroidectomy and ablation with iodine-131 and thyroid hormone replacement therapy. And at high doses of sodium acetate, 50 megabecquerel per kilogram, transient bone marrow suppression and pathological changes in the testis were observed. So it should be paid attention in the clinical trial, which is scheduled next year. So this is a roadmap for clinical trial. So we finished preclinical examination and toxicity study, and we are now consulting PMDA, Japanese Authority for Drug Approval, and hopefully we can start clinical trial next year for the refractory thyroid cancer patient as a phase 1 clinical study. So so astatine was provided from the Research Center for Nuclear Physics at Osaka University, RIKEN, and QST via short rapid RI supply platform. So I'd like to acknowledge for the involved researchers and staffs in these facilities. So and finally, I'd like to thank my collaborators, especially Dr. Shirakami, Dr. Kaneda, and Dr. Toyoshima in the Institute of Radiation Science. So thank you very much for your kind attention.
So thank you for this kind invitation to present on this Warmth 2020 conference. I'm Øyvind S. Bruland. I'm a clinical oncologist. I'm sitting here with Asta Juzeniene, who is a physics radiobiologist, and we will share with you some of the preclinical platform of this dual alpha targeting platform, which we have presented in the abstract. It's actually a novel technology platform, and we will share with you the data we have in prostate cancer models. The patents are owned by the small startup company Nuclegen AS Incorporated, and we are aiming for first-in-man studies already in 2021. I have to say that the Oslo Group has an extensive experience in developing therapeutic radiopharmaceuticals. Together with Roy Larsen, I was one of two founders on the Algeta Bayer Sofigo story, which most of you know. We have already also been in the Nordic Nanovector. It's a beta-emitting CD37 targeted radioimmunotherapy. And last but not least, the OncoInvent company is well into phase 1 clinical trials. And the reason why I show this is because here we also build upon the alpha emitter radium-224. And we have shown in the patients, 15 patients so far, that the daughter isotope, lead-212, can be used as a imageable indicator of how this radiopharmaceutical distributes in the body of real patients. And we hope that will be the same in the story we will tell you about now. The aim, as you see here, is to utilize as much as the possible of the energy from the entire decay scheme of chain of radium-224 all the way down to stable lead, and target as much as possible of the energy of ionizing radiation from this decay chain to tumor sites. Most of you know that at present, patients with late-stage metastatic prostate cancer failing available treatments now have both bone and, to a larger extent, extraskeletal metastases that are associated with poor prognosis and decreased quality of life. And there is definitely a need for novel treatments to combat the entire spectrum of metastases. That said, bone metastases are still very important, but the patient needs something more to combat visceral metastases and also lymph node metastases. And I know that you have heard earlier today a lecture on lutetium, and you also have the theranostic and the PET PSMA-based new diagnostics. The dual targeting platform opens for several product candidates. It can be the volume seeker radium-224, which resembles the 223 Xofigo, but with a shorter physical half-life. It can be the lead daughter can be targeted to the chelator EDTMP to get a bone surface bone seeker together with a volume seeker. And Asta, you published with Roy a paper on that 2 years ago, I think. But can you go further here? Because there are other options where this daughter can be via a chelator coupled to, for instance, monoclonal antibodies, oligopeptides, other chelator conjugated targeting carrier molecules. And it has to do with the ingrowth of the daughter.
This is very correct. I just want to mention what we cannot use radium-224 directly to chelate, because not radium-223, not radium-224 has possibility to be chelated due to the lack of chelators. We cannot use radium-223 because he has a daughter 211 lead, which has only 1 hour half-life. In this case, we have 10.6 hours. Lead is a beta emitter, but its daughter bismuth is alpha emitter. Why it's possible? In the beginning, we have radium-224 alone only. And then time goes, radium-224 decays, and its daughters appears. After 2-3 days, we have equilibrium situation. Then we have radium-224 and lead at the same time. To this solution, we can add, as Øyvind mentioned it, different targeting molecules, including monoclonal antibodies, PSMA ligands, and the process is very fast. Labeling is very fast. I will talk later in details. Then we can get 2 radionuclides in radium-224 solution.
So an example, as Asta mentioned, the bone seeker radium will target bone metastases if they have the sclerotic stroma, which is well known to you from the Xofigo story. But in addition to that, for instance, in prostate cancer, if we then utilize a small ligand to the to the cell surface marker PSMA, we can also target the indirect alpha emitting daughter 212 lead when it decays to bismuth. It will also deliver that alpha particle to the specific individual cancer cells. This target is well known to the audience. I think you have probably in the conference seen a lot of presentations on PSMA PET imaging. It has revolutionized the selection of patients, for instance, to select the best local treatment, being surgery or oncological treatments before definitive radiotherapy, for instance. But for this purpose, I think it's very important to mention that it is regarded as an internalizing antigen. And that is important when the daughters are, when lead is transported into the cell and the daughter bismuth gets closer to the DNA and the cell nucleus. Some of you may have seen the two papers of our PhD candidate, Vilde. We will share with you just very few of the results related to synthesis of the novel ligand NG001, where also Nucligen has acquired the patent, originally developed by Roy Larsen, the nuclear chemist. We will go through the novel liquid radium lead generator for preparation of dual alpha targeting solutions, and should share with you some results from in situ, in vitro, and in vivo results. And also we have compared to 12 lead labeled NG001 with the well-known ligand 617 PSMA, also with 212 lead labeling. Will you comment on that, Asta? The NG001, it's a more lipophilic variant.
Yes. As you see, we have the NG1 and standard PSMA 617. Both of them, they have chelators. This is standard chelator daughter. And in our case, we have TCMC, which is much better to use with lead-212. Binding motif for both compounds are the same. However, linkers are different. Due to this, our compound has a higher molecular weight and is more lipophilic. In this case, we have less lipophilic and smaller molecule. Both parameters can influence cell binding and distribution in tumors.
And also in the body of mice.
In the body, it can be related to toxicity, and we have studied and compared both.
And also renal excretion, etc. We'll come back to that.
Excretion. Which of them are best? And as Øyvind told, we have developed a new radium-225 liquid generator. Why this is important? Because radium-224 solution can be delivered to any site, and labeling can be performed just before injection. And then we make radio labeling. We can use radium-224 solution already containing lead, ligand, or we can purify. This is important because this is increase transportation. We can use this product free for days, which makes much easier. If to use only pure lead, it must be transported within 1 day and used. The process of radiolabeling is very, very fast. As we earlier showed, radium-224 solution must be in equilibrium with daughters, including lead, and we add ligand, wait 40 minutes at 47 degrees, we don't need to heat, and when we have the product, radiochemical purity was achieved over 90% at concentrations above 20. As you see, we can go for very different specificities. If we need, we can purify it with quite high yield, 85%. We also tested both compounds, both ligands, NG001 and PSMA, and here the data presented for cell surface bound, and we see what we don't have difference in vitro for binding and internalization of both compounds. Green area presents clinical relevant doses where we have very high cell surface bounding and internalization. We also tested purified radioligand, our ligand, in spheroids. And when we add clinically relevant activities, 3 and 5 kilobecquerel per milliliter, we see that after 4 and 48 hours, we have very strong cytotoxic effect. In the control group, we pre-blocked receptors with cold ligand, and what we see, what any cytotoxic effect was observed. What means what radioligand must bind and maybe be internalized in order to be cytotoxic. We also studied biodistribution of lead and radium of 3 different compounds. Free lead, as you see, label it with Ng001 and PSMA-607 in the presence of radium-224. Drugs were injected intravenously, and after 2 hours, mice were autopsied. And we measured it, biodistribution of lead and radium-224 in different organs. As you see, both radioligands were cleared very, very fast from the blood. In tumor, we had much higher activity, percentage of injected dose per gram of both products were very similar after 2 hours, while alone freely had very low. If we look for kidney uptake, the lowest uptake was observed with our radioligand, Ng001, 3.5 times higher had PSMA-607, and free lead was in the middle. I also want to mention what our product was very stable because we haven't observed very high activities in bones, femur and skull, while free lead had quite a high activity in bones. Then we look for radium-224. We see that all 3 solutions had similar biodistribution in bones, which means what radium-224 can be used and we don't change, labeling doesn't change properties and high uptake. We also tested pure lead-200 G1 at different times, and we see what it's very high uptake and very fast uptake in tumor, which lasts around 24 hours, while it's very fast cleared from the blood. In kidneys, we have very high uptake in mice, but it's cleared very, very fast. We need to remember what kidneys of mice have much higher number of antigens than of PSMA receptors than humans and cannot be directly transformed to this data. At the same time, we see very fast clearance, and around 3 hours, we have very similar data for both organs, and then only decrease the highest we have in the tumor. Bismuth is a product of lead which can be released during lead decay and can be relocalized from target organs and can cause toxicity. And we tested biodistribution of lead and bismuth in different organs, and we see what uptake values for both products were very similar in all organs, and spectra you can see here indicated what product was very, very stable and no extensive translocation of bismuth from lead in other organs.
So then it comes to sort of proof of the pudding, because what about therapeutic effects in murine models of prostate cancer? And here I should stress that so far we have only been able to study in more detail the single alpha part of this dual alpha, because to our knowledge, there is a lack of prostate cancer animal models with sclerotic stroma or osteoblastic stroma within the metastasis. So it's very difficult to study. We have shown uptake of radium in bone, but we do not have an osteoblastic prostate cancer, either subcutaneous or metastatic model, that can allow us to study effect in a human prostate cancer cell model in mice.
Having PSMA expression.
And simultaneously with PSMA expression. But I hope you agree with me that there is actually very much known in the clinical real world on radium biodistribution in patients, both with the Xofigo development and also the historical data from radium-224 in ankylosing spondylitis, both as bone seekers. That was done decades ago in Germany, and there has been a couple of publications on long-term outcomes, secondary cancers, leukemia, small excessive osteosarcoma. But we focus on patients with very poor prognosis from end-stage prostate cancer. So we don't think that will have any relevance. So the therapeutic effect I'll show you has been done with purified 212 lead Ng001. And here, Asta, we use the C42 model.
PSMA expressing human prostate cancer cells, resembling very much human cancer.
Yeah, and the mice were treated with 0.3 megabecquerel, which was at or below the maximum no observed adverse event level in mice. I'll come back to toxicity later.
Because we have not tested at high activities which can be maybe toxic or not toxic, but this data we are now
working on for a publication.
And we are not toxic.
Yeah, and we cannot share with you all the details because it has not been published yet. But here you see, compared with control, there is a significant stabilization or prolongation of low tumor volume compared with the rapidly growing tumors in the control group. And that translated into an improved survival outcome of mice in the 001-212 lead group compared with control. Median survival at 0.3 was 38.5 days, and we calculated a therapeutic index of approximately 2.3. We have also, here you see conventional BALB/C white mice. We have studied short-term and long-term toxicity compared with saline intravenous injections, small activities injected, and also larger and repeated amounts of injections. Would you expand on that, Asta?
Yes, I think we can go directly here because the results are very clear, because we have not observed any toxicity. Body weight wasn't changed. This is months. You see, we followed all these animals 1 year, and we have terminated at 6 months and 12 months, and we sent all organs to pathologists. I will show later a little bit details, and we measure it every month, blood parameters, full blood hematology, but we haven't observed any changes in blood. And as I mentioned it, organs were sent to pathologists, and we haven't found any difference, any alterations in any of the group.
And particularly not any kidney damage as evaluated by the pathologist.
And we also weighted organs and we did more tests, but we haven't observed. At the present moment, we have increased activities and doing new studies to find
maximum tolerable
dose in animals.
So in summary, I think it's fair to say that the PSMA ligands were efficiently labeled with 212 using the generator system and showed high binding and internalization in PSMA-positive cells. Regarding biodistribution, this novel PSMA targeting ligand showed favorable biodistribution and similar tumor uptake as the well-known 617 ligand already after 2 hours, but with a 3.5 times lower kidney uptake, which we think is a significant finding. Regarding therapeutic effect, tumor growth delay was observed for this radio-labeled ligand at relatively modest activity doses, and improved median survival with the therapeutic index in the various studies between 2.3 and 2.7. In one study, it was 3.5.
It's different because but it's because we made many studies.
Okay, and regarding toxicity, both the low and the high and the repeated exposure of this was well tolerated without any signs of long-term organ toxicity. I think that was the end of our presentation. Thank you for listening.
Thank you very much.